Okay, this segment is going to focus on lipids, our macromolecules lipids. And just a reminder, lipids are your fats, they are your waxes, and they are your steroids. So when we look at a lipid and we look at the diagram, typically what we're looking at are your fats. Okay, your waxes and your steroids tend to be cyclic structures. So when we're talking about lipids, we are probably most of the time talking about things that look like that picture that you see there. Basically, what we have going on here, just as a reminder, the signature group here are these esters. Remember, they're what tastes good. And we're typically looking at a big, long fatty acid chain. So let's take a look at what a fatty acid chain is. That's our monomer for these guys. So originally, what a fatty acid chain looks like is your carbon. And how do we get it called an acid? Well, we put carboxylic acid on it. And then we start attaching the fatty part. Basically, the fatty part is coming from this very long hydrocarbon chain that's going to sit over here. So this is what we refer to as a hydrocarbon chain. If we did a condensation reaction, this could possibly be the hydrogen that comes off. And what you see sticking out over here, this guy here, that long stick piece right there, that is called glycerol. So technically we would need 3 of these to satisfy these guys. And before we did our condensation reaction, we had 3 OH groups hanging off of there. When we got rid of those, we got these esters. And that's what those bonds are, remember. Here's our ester. Remember, Uncle Ketone married Aunt Ether, and they had baby Esther. So we have 3 ester bonds there. And those are that's basically some general information about our lipids and our general way that we make them. I want to clear the screen here, but I don't want to get rid of everything, but I think I'm going to have to. And let me put our picture back up. Okay, here he is again, because I need to talk to you about the chains themselves. we take a look at this chain, and we take a look at this chain, we can see that all of the carbons have as many H's as they can hold. They are therefore called saturated. These two chains are saturated fatty acid chains. Whereas, on the other hand, this chain, well, let me actually draw it where it's supposed to be. Hang on there, guys. This chain here has one set that are double bonded. To each other. That makes that chain unsaturated. So when you hear somebody talk about a saturated fat, that would mean all three of those chains would have to be single bonded and carrying as much hydrogens as possible. As soon as one chain becomes double bonded between two carbons, then you now have an unsaturated fat. All it takes is one set of carbons. But typically, we're going to be looking at fatty acid chains that are 30, 40, 50 carbons in length. So there's going to be many double bonds in an unsaturated fat. And in a saturated fat, that's really bad. You're going to have all of those big, long carbon molecules totally filled with as many H's as possible. And that's our little tidbit about our saturated and unsaturated fats. Okay, so this image that you have is found next in your notes, and this is probably one of the most important lipids to us in biology. This is called a phospholipid. So it's still a lipid. Here's our original glycerol that we were looking at when we were looking at unsaturated and saturated. We have two fatty acid chains. But what makes this different, let's see if I can get rid of some of my circle there. Oh, well, that's not what we wanted to do at all. What makes this different is this group here that's a phosphorus with 4 oxygens. That's the phosphate group. Thus, the name phospholipid. So, phospholipids are found in your cell membranes. We're going to spend a lot of time with them in the next couple of chapters. They exist in a bilayer. And the interesting thing that happens with the phospholipid, we usually draw it looking like this. with its 2 fatty acid chains. So these are the fatty acid chains down here. And then this is the PO4 grouping up here. This becomes charged. So this is a polar region. And these guys down here are nonpolar. So it's an interesting structure because you have this polarized region up here, and then you have these tail regions down here that are nonpolar. So if they sit in the bilayer, that's what they would look like. So the 2 nonpolar regions are facing each other, and the 2 polar regions are opposite ends. And this becomes important because out here, there's a lot of H2O. And inside here, there's also a lot of H2O. And hopefully you're remembering from the basic chem chapter that water is a polar molecule, and therefore these 2, polar and polar, will get along. So the structure of this phospholipid allows for its function. We're not going to spend a lot of time on that right now, but it is worth mentioning that this is the first particular case where we're seeing the actual structure of a molecule that allows it to function within a living substance or a living organism the way it does. All right, last thing I want to talk about before we're done with our lipids are the guys that aren't your normal lipid shape. So what I got for us was this picture up here, which is a bunch of steroids. As you can see, a lot of them are hormones. Hormones are natural steroids. So you have testosterone in here, progesterone in here. Cortisol is a natural hormone. So is aldosterone. They all are. So hormones are steroids. And steroids are lipids. But as you can tell, they're not your fatty acid chains that we have just finished looking at. So these guys are definite ring structures. And again, they can look, pretty similar. They're just these ever so slight differences in them, especially if you take a look at this ring and then these guys and then this ring and then these guys and then this ring here and those guys. They're essentially all the same. Same thing with this guy down here and this guy down here. So cortisol, corticosterone, aldosterone, progesterone, and testosterone. If you look at those structures, these guys here, those rings, they are essentially the same. Okay, now, we can't say they're the same chemical formula. They're not isomers of each other because you have an extra OH group here, whereas here you don't have an OH group. It's missing. Here you have this thing, whatever it is. It's actually an aldehyde sticking out there. That's how you get aldosterone, aldehyde, ALD in the front of it. This guy here has none of that. He just has this ketone here. This is a ketone here. That's a ketone. Here we have, well, we don't have the same ring, so that doesn't count. But here we have just an alcohol group hanging out. So they aren't isomers, but they are very similar in their structure. And because they are those rings, again, remember those ring structures, they get pretty hard to break. So a lot easier to break a fat down than it is to break these guys down. But one of the things we do need to talk about is that these guys are, they tend to be what you hear them fat soluble. Sometimes vitamins are said to be fat soluble. It means that your fats are going to store those substances. And so sometimes that's not the best thing going on. This structure down here, he's cholesterol. So again, just taking a look at your structures, you still have, 3 cyclics that are 6-sided and 1 that is a what is that called? 5-sided, whatever that's called. I want to say it looks like a house, but I'm sure you're all laughing at me now because it doesn't. That's not what it's called in geometry, I'm sure. So one of the last things I want to talk about is you hear about good cholesterol, and then you hear about bad cholesterol. And in your notes you have a picture of, the quote-unquote good cholesterol and the bad cholesterol. And the good cholesterol has next to it the letters, or it gives you the image of an angel wings, and that's HDL. And then you have bad cholesterol, which is given to you pictorially as LDL and the little devil. HDL, those letters stand for high density lipids. And LDL stands for low density lipid. And the key to understanding this is this word right here. So something that is got high density. Remember what density actually equals. Density is mass over volume. So something that has a high density has a large mass but a small volume. Something that has low density has a smaller mass and perhaps a larger volume. Not necessarily, but it could be. So if something has a high density, in your heads, I'm seeing, or I'm thinking I'm seeing, that you think that these are heavy. And then these would be light. And I want you to think about that and think about where cholesterol is associated. We always hear, or you should hear, hopefully, that people have their blood checked for their cholesterol levels. And they look for having low HDL and a higher or more acceptable range of LDL. You need to have cholesterol inside your body because cholesterol in general is part of the cell membrane. So cholesterol is important to us. It's not that we don't want it in our diet at all, but we want to make sure we have the right kind. So this HDL that's heavy is no good because, or pardon me, is good. Let's start over again. The HDL that's heavy is good because it's not going to travel fast. And as it travels through your blood system, it actually does a pretty good job at picking things up and moving them along because it's not traveling very fast. This guy, on the other hand, the low density, travels very fast. And because he travels fast, he knocks into things. crashes. And because he crashes, he sticks. It's this guy who's going to be clogging up your arteries. That's a lot of information to squeeze on that page. But that's the difference between good cholesterol and bad cholesterol. Those heavy guys, they're slow movers. They'll move along. They'll even pick up things and take them with them, kind of getting rid of stuff for you. That's a high-density lipid. On the other hand, those low-density lipids, that version of cholesterol, not good. He's light. He knocks into things. He crashes, and because he crashes, he's going to stick to stuff. So that ends the lipids portion. So now you can move into doing some of the reinforcement work for carbohydrates and lipids, or you can move on to the proteins notes.